Webinar. It's a real pleasure to have Professor Hanspeter Herzel here again with us after 5 years. And sorry again for the confusion with the time zone. From now on, I'm going to send Outlook invites. It's my fault. Professor Herzel is a professor at the Charité, a professor of theoretical biology, and this group has been established since 1996, I believe. He did his habilitation in Humboldt University in 1990 after a PhD in theoretical physics. And then he has been doing seminal work in nonlinear dynamics and oscillators. And we are very happy to hear his talk on oscillator theory means biological rhythms. Over to you, Professor Herzel.
Okay, thanks for the organization of this meeting and sorry for the delay. But in fact, an hour ago, I gave another lecture here at EMBL, since we have the fourth oscillator meeting. And some of you might remember these meetings. So Leon Glass and Michael McKee were attending the last meeting. So it's again fun. But I'm also happy to be back in Montreal, since I had also nice hours, days, weeks with Leon Glass and Michael McKee and on Makadra in previous years. So it's a pleasure to come back, at least virtually, to Montreal. So the idea, so I scanned a bit the audience, and I'm aware that some of you are mathematicians, others are biologists. So I tried to I collected material from biology, from mathematics, with a couple of take-home messages. I'm happy to answer questions in between, and I will stop after, say, 45 minutes and go step by step from one story to the next. Okay, of course, the work is done together with a couple of great colleagues, and some of them are listed below. By the way, Samuel Bernard got his PhD from McGill. So now I try to go to the next slide, and I fail.
Oops, any help, Alex?
Alex may have stepped out. Hold on, let me just go see.
Maybe stop and start again.
Yeah, maybe stop the share and start again.
Stop share. Share screen.
And then just make sure you stay in your PowerPoint to be able to change the slides.
Now it works.
Excellent.
Okay, can you see the next slide outline?
Yes.
Okay, I start with biology, so I simply list a couple of examples. Then I give some general principles, and if time is left, then I go to my favorite current topic, to the circadian clock. So this is a nice sketch of oscillators and conservative and dissipative systems from a nice textbook. And you might see already some biological oscillators. So in the middle, there's a sketch of the circadian clock. There's a heart, and there are biological and engineering and physical examples of oscillations everywhere. And the theory is very interesting, but not easy. So you include CAMBAM theorem and so on and so on. But I personally learned a lot from a beautiful book of Michael McKee and Leon Glass, From Clocks to Chaos. And the theory was known to me, but I learned also a lot about physiology. And here's one of the most impressive examples. Say they studied single cells, heart cells, and were pacing them with electrical pulses. And then they got an empirical response curve and simulated the response curve and predicted there might be one-to-one locking and one-to-two locking and two-to-one locking. And then they found examples in the recordings of all these locking regions. And this is at the end perhaps the most complicated slide of my talk. So it's a two-dimensional bifurcation diagram combining data and theory. And now I will become a bit more simpler, going to other examples. This is say another example of self-sustained oscillations. These are so-called central pattern generators, or CPGs. These are networks of cells, like our respiration clock. This is a network of inhibitory neurons. And in this system, you can also see a lot of rhythms. And if you change parameters, then you can change from no oscillation to oscillation. And this is termed bifurcation. And if the oscillations are self-sustained, you call it limit cycles. And this is a slide from my own research. So in the early 90s, we studied voice examples and found regular voices like ee, or chaotic voices like ah. And these are data from patients. And what I imitated was more or less chaotic voices from left and right vocal fold. I could somehow drive into subharmonics and chaos. And together with Ingo Tietze in Iowa City, we also simulated these voice, and every voice signal in vertebrates is driven by air, and there's something vibrating, typically vocal folds or syringes in birds. And this vibration can be regular, as shown in the middle graph, or chaotic, as shown below. And you can understand it using simulations and mode analysis and so on. It's just a snapshot from another interesting field. And in my eyes, if you ask me where nonlinear dynamics was particularly successful in the last decades, then I would say in heart arrhythmias, thanks to Leon Glass and Michael Guevara, and also in voice research, thanks to Ingo Tietze and other friends. And this is a bit more recent. I learned from Tecumseh Fitch that also in the animal world, chaos is not the exception, but quite frequent. And here you see 4 calls in the spectrographic analysis. The first call is quite regular. You see a fundamental frequency of about 400 hertz and a lot of harmonics. In the second call, you have a bit of subharmonics at the beginning, and in the last call, you have a lot of chaos. So even a single individual can switch from regular phonation to chaotic phonation. Now I move to more molecular clocks, since I'm aware that most people these days are interested in genes and regulations of genes. And here you see again 6 examples from, say, cellular or genetic oscillations. My favorite topic is the circadian clock. So they are activating transcription factors like BML1 and clock, and there are inhibitors like PET and cryptochrome. But also the somite oscillator is very interesting. So there are oscillations of, say, 40 minutes in zebrafish, of 2 hours in mouse, and of 5 hours in man. And these oscillations are really essential for early development. So it's also physiologically very, very important as a circadian clock or heart rhythms. The other examples are less clear what they mean. There are oscillations in p53 oscillations due to an inhibitor, MDM2 and E3 ligase. There are oscillations in NF-kappa B cells. There are glycolytic oscillations in metabolism and CAMP oscillations. And all of them have some functions, but the obvious biological function is sometimes missing. That's why there's a lot to do, even if you see oscillations, to understand the evolutionary role. Now I come back to the mathematics. So first of all, here are some heroes of clock modelers in chronobiology. Rüdiger Weber, Brian Goodwin, Art Winfrey, I met in Montreal at the conference, and so on. John Tyson, still very active. And all of them explained that in order to get self-sustained oscillations or even damped oscillations, you need delayed negative feedback loops. And the simplest equation is a one-dimensional delay differential equations. I personally learned it from Michael McKee, that this is a very powerful system. And if you understand the upper figure, then you might learn that there is a close relationship between the delay, term capital T, and the period. So The short summary is the delay is a fraction of the period, typically between 1/4 and 1/2 of the period. So in other words, if you know the period, say, of 2 hours in mouse stomate oscillations, then you can predict the period, the delay of about 45 minutes. So it's very helpful to have this rule in mind that a longer delay gives a longer period. And this leads me to an interesting biological question. In the mammalian clock, we know the period very well. It's about 24 hours. And the theory predicts we need at least a 6-hour delay. And this is by no means obvious how a transcriptional translational feedback loop can lead to a delay of at least 6 hours. There are some preliminary answers. We know that multiple phosphorylations matter. We know that big complexes are formed. We know that in some of the proteins are intrinsically disordered, and this can all contribute to long periods. And also epigenetics is involved. But there is no final answer how the period can be controlled in such a relatively precise manner. So in summary, from a really textbook story that delays and periods are related, you can easily go to the most advanced questions in chronobiology or even in stomate oscillations. Where are the delays? Where are the nonlinearities? How to get self-sustained rhythms? And now I switch to my favorite topic, to the circadian clock. So all of us know the Earth is rotating with a period of 24 hours, but not everybody is aware that in many, many organisms, from cyanobacteria to Neurospora, flies, and mammals, there's an intrinsic clock. And this clock is really pronounced. And here I show some details. So it's visible in physiology, sleep-wake cycles, for example, or body temperature, but it's also visible in the brain. And now I have some battery problems. That's unexpected. Now it's gone, sorry. And coming back to the circadian clock, the clock is ticking on the organismic level. So on the left-hand side, you see that hormones like melatonin and cortisol are oscillating, but also body temperature is half a degree deeper in the night in us than during the day. In mice, it's even two degrees. And interestingly, all these rhythms are orchestrated by a tiny brain region termed the suprachiasmatic nucleus. And the trick is that this is one of the few brain regions that have direct light input. And that's why the light dark cycles can trigger the rhythms or control rhythms in our brain. And these are at the end transmitted to hormones, to physiology, to the body. And from a mathematical point of view, it's interesting that on the one hand, the rhythms are generated in single cells by negative delayed feedback loops of some core clock genes. But in our brain, we have 20,000 neurons, and they are coupled very well with VIP, VPEG2, and this leads to synchronization within the suprachiasmatic nucleus. Moreover, these regions are driven by light. So we have, in addition to the intrinsic oscillators that are coupled and synchronized, we have also the extrinsic day-night cycle. And this is a network of coupled oscillators, and there some theory is helpful. And this is a summary of the circadian oscillator. So I start with the behavioral rhythm. So you see a hamster, and if the hamster is driven by light dark cycles, then you have little activity during the day and say pronounced activity during the night. These are the black regions. But the fun part comes after a week. So then we turn off the lights, and this is termed DD, dark dark. And then still the hamster is precisely rhythmic. So, but he starts to run every evening half an hour earlier. So we can read that the period of a hamster under free running conditions is about 23.3 hours. And this is interesting since it's a bit shorter. And that's why the circadian clock is about a day circadian. And in the 80s, 90s, all the genes have been discovered that are responsible. And they are termed BML1 or period or cryptochrome. And you see here some sketches of mRNAs. They peak typically during the day. And you see the associated proteins. And you see immediately that there is a delay between mRNA and protein levels. And this is a hint that there is indeed enough delay to get a delay of 6 hours and a period of 24 hours. And there are even some mutations, familiar advanced sleep phase syndrome, for example. You have a point mutation in a phosphorylation site, and this leads to a shorter delay and a shorter period. And these poor people, they go to bed at 8 o'clock in the evening and get up at 3 o'clock in the morning. So it's really an exciting example from a point mutation leading to a very strong phenotype. And in the meantime, we understood many, many details. That the lifetime of the PA2 protein is changed from 6 hours to 3 hours and so on and so on. So it's a lot going on, but it's still understandable in the framework of self-sustained oscillations of delays and period and phase control. And this brings me to my favorite level, to the modeling level. All this can be modeled with simple differential equations, with X, Y, Z models with delayed negative feedback loop, including some switches. So we need nonlinearities to get self-sustained rhythms. Okay, and say a few years ago, Michael Rosberg, Jeff Hall got the Nobel Prize for the discovery of clock genes in Drosophila. And the discovery of the genes was already in the 84, but this paper that is sketched here in this figure was from 1990. And here Michael Rosberg realized that period can inhibit its own transcription. And that's why this was the first paper where gene regulatory feedback loop was claimed to be the source of self-sustained oscillations in single cells, in this case in Drosophila. In the meantime, we have also single cell rhythms in mammals, and similar genes are involved. And here's back to mathematics. This is a very specific delayed negative feedback loop with the period gene, and you see again, if there's a delay and some nonlinearity, then it's easy to get 24-hour rhythms in a simulation. And if you go to the lower right graph, then you see the delay, the orange line is controlling the period. So there's a clear relationship between tau, the delay in this case, and the period length. And of course, for the proper delay, you get a 24-hour period, at least in the computational model. Okay, this is the first sub-summary. So it's true that in order to get self-sustained oscillations, you need delayed negative feedback loops and some nonlinearities. And there's a clear relationship between period and delay. And this helps you to interpret biological data. The question is where the delay is coming from. And at least in this OBEC expression essay of Michael Young, another Nobel Prize winner, it is clear that driving proteins, period and timeless, Per and Tim, have a 6-hour delay. And this, I guess they made this draft even without knowing the mathematics of delayed negative feedback loop. It was just an observation that it takes about 5-6 hours for this dimer to import to the nucleus. And in fact, this Achim Kramer, my biochemistry colleague, we tried to reproduce these findings in mammals, but say in mammals, nuclear import is too fast. So in mammals, we need other mechanisms like phosphorylation of IDPs or complex formation or epigenetics. So it's still a hot debate what gives us a 6-hour delay in the mammalian circadian clock. But it's at least helpful to know the theory predicts a 6-hour delay. Okay, so circadian rhythms are generated by positive and negative feedback loops. The positive feedback loops are helpful to get nonlinearities. Long delays are essential, but the mechanism is still debated. And complex nonlinearities are essential as well, but here also different mechanisms might contribute. This is also an ongoing discussion. What is the real major nonlinearity? So now I have time to go on with the next story. So we learned also from experimental data that single cells are rhythmic but sloppy. So here you see the period distribution of single cell data, and you see it's between 20 hours and 28 hours, and quite distributed and quite noisy. If you make a brain slice, then you can already standard deviation of half an hour. But if you look at hamsters or mice, then you get an accuracy of plus minus 12 minutes. So it's really amazing that the period control is very, very strong. And this is obviously due to coupling. So you have, in addition, you couple, say, these 10,000 neurons via neuropeptides, and then you get much better rhythms. And that's why we asked ourselves, what about the single cells? Are these good oscillators or sloppy oscillators, damped or self-sustained? And here are some data we got from Okamura and David Welch. And you might ask yourself, are you safe that these oscillations are limit cycles, or could it be a weakly damped oscillator with some noise? And the answer is non-trivial. And if I go on, then I can show you even better data from Tanya Laisi and David Welch. And they found the oscillations over 42 days in single cells. Still, it looks a bit like a limit cycle, the best cells, but we asked ourselves, can we simulate these cells with damped or self-sustained oscillators? And this is a bit of mathematics. So the upper model is just a damped oscillator driven by noise. And the autocorrelation functions can be calculated analytically. And the lower model is a linearized limit cycle oscillator. You see the capital A, there's an amplitude. There's an amplitude relaxation rate, a Fourier exponent, lambda, and there's again some noise. And thanks to Stratanovich from the 50s, he can also calculate the autocorrelation function and compare it with the data. And then our shock was that both models work. So we studied all data we could get, single cell data, and you see some experimental data on the left, but you see simulated damped oscillator, simulated self-sustained oscillator, and the properties are a bit different, noise levels, but say it's hard to distinguish from correlation functions or other fits these data. So at the end, we have to conclude, say, single cell mammalian cells have a clock, but they are close to the Hopf bifurcation, close to the onset of oscillations. Since they might be either very weakly damped and noise driven, or they are limit cycles, but close to the Hopf bifurcation. And this was a bit disappointing at a first glance, since we all love limit cycles like Van der Pol oscillator, Goodwin oscillator with very strong rhythms. But it turned out weak oscillators have also the advantage, since they lead to less chaos and are more easily synchronized and entrained by external inputs. So at the end, I'm now enthusiastic about weak oscillators since they are more brave. They show less chaos and are more easily to synchronize. And this is illustrated in the next 3 slides. So if, first of all, we have weak oscillators, but if we couple strong oscillators, here is a Goodwin model with a mean field coupling, then we get chaos, tori, and even steady states. So by simulating two cells that are strong limit cycles, we get quasi-periodicity, synchronization, PA doubling, and chaos. So that's what I expected. But if we simulate 100 cells, sloppy cells, then synchronization is easy. So this was done again with Samuel Bernard from originally Montreal. And he has shown if the oscillators are damped, so it's seen in graph, say, F or E, that if you turn off coupling, then it's becoming a damped oscillator. So, but damped oscillators are easy to synchronize. That's why at the end, I'm no longer disturbed by the fact that our oscillators are sloppy, perhaps even damped, but it's at the end a design principle. So if you want to drive, say, oscillators, if you want to synchronize them, then it's good to have sloppy oscillators, then chaos is less likely. And that's also the observed effect. So in rats, mice, in say more than 90% of all brain slices, you find well synchronized rhythms. So chaos is the exception, even though the P had spread a lot, and this supports the sloppy rhythms in single cells. And this has been measured also by Eric Herzog, my colleague and friend, and he found out that most single cells are quite noisy, damped or unstable. And this is consistent. And this is an interesting design principle that, say, we can distinguish weak and strong oscillators. And strong oscillators are good to have stable, robust amplitudes, but weak oscillators are better to be synchronized by the environment or by coupling. Okay, so my second summary is that indeed single neurons might be noise weakly damped oscillators, but they are easy to synchronize or to tune to the environment. But coupling strength is also important. And I feel there's still a bit of time for another story. So, but perhaps it's also time for questions. So if you have questions right now, so then it's also time to go on with some discussion.
Anybody has a question, you can put it in the chat, or you can sort of just turn on your microphone and ask your question right now. Andre.
Yes. Hi, Hanspeter. Nice to see you again. Thanks for the talk here. When you say are easily synchronized and entrained, do you mean by each other or by really the external sun rhythm?
So in this case, we simulated slices, and slices are internally synchronized. So, and we played around with local coupling, we played around with long distance coupling, we even added heterogeneities like right dorsomedial and right ventrolateral. And in all cases, we could reproduce the observed synchronous patterns with some phase waves. So in this case, I mean synchronization only.
Synchronization between each other,
Yes.
or to an external rhythm?
No, between each other. No? In my final part, no, I will go to the entrainment. So we decided to call, say, cell-cell interaction synchronization and sun clock synchronization entrainment. This makes it a bit simpler. So if I use the term entrainment, then I have a Zeitgeber in mind and intrinsic clock. If I speak about synchronization, then I speak about cell-cell synchronization or the liver brain synchronization.
Right. So the quasi-cycle makes it more malleable in a way, or more flexible to be synchronized with others.
It's a take-home message that weak oscillators are helpful. This applies to both. So we synchronize, for example, with mean field, and the mean field is a kind of external oscillator at the end. Even it's self-made. So it's a self-made neuropeptide oscillation. Okay, other questions, comments? Okay, then I go on and come to the issue
There is
of
one more
entrainment.
question. There's
Yeah.
one more question from Katie.
Mhm.
Hi, sorry, it's just a quick question following on the previous question. When you say that the cells are easily entrained or synchronized easier, is that also independent of the frequency of the stimulus, or like would it be more likely to synchronize with slower frequencies to its own or faster frequencies?
So it's also related to frequencies. So let's say, for example, in mammals, we have a strong clock in our brain, the whole SCN. And that's why we can entrain to plus minus 2 hours. So it's easy to entrain mammals to 22 hours or 6 hours, but impossible to entrain to 20 hours and 28 hours.
Okay.
But in Neurospora, this is a single cell oscillator somehow. Ne? There we can entrain also to 20 hours or 28 hours or even to 16 hours. That's why your intuition was right. Weak oscillators are not only robust or do not show chaos, they are also entrainable to mass periods or to long and short periods. Whereas strong oscillators with large amplitudes or strong coupling, they have a small entrainment range.
Okay.
And if you ever heard about Arnold tongues, this is the visualization of this property. So for strong oscillators, we have a narrow entrainment range. For weak oscillators, we have a broad entrainment range. For a given Zeitgeber strength. Of course, we can also go for strong Zeitgebers, and we have a slightly broader entrainment range. But some of these issues reappear in my third part. But first, let's agree about what we call entrainment. So we have an external rhythm, in this case, warm-cold cycles. In physiological situation, the light-dark cycles. And we have an intrinsic period, and the period becomes equal to the Zeitgeber period. So physicists might call it frequency locking or, or phase locking, but we call it entrainment. And interestingly, if there's frequency locking, then typically there's also a well-defined phase between extrinsic clock, so the sun, and us. And that's why some of us have an early phase, morning larks, and many of us have a late phase, night owls. And this is physiological. And I could even show data. So these are data from the intrinsic clocks of humans. And you see the period is a bit longer than 24 hours in humans, at least in young students in Boston. And but the period is extremely precise. So it's plus minus 12 minutes. If you measure the chronotype or the, say, mid-sleep time, for example, or the dip in body temperature or the onset of sleep or the onset of melatonin, then you get a much broader range of about plus minus 90 minutes. And this raises a straightforward question. If our clock is running 12 minutes faster, why do we get up 90 minutes earlier? And this is not intuitive, but physicists understand the answer quickly. So it's related to strong and weak oscillators. But I will come back to this question in a minute. Let's first
There's one more
illustrate.
question in the chat. There's a question from Victoria. Is a weak oscillator the same as damped, or is it a broader term?
It's a broader term. So I would call a weak oscillator definitely as a damped oscillator definitely as weak. But would say a small amplitude oscillator might be also termed weak. And you can also include the amplitude relaxation rate or the Floquet exponent. So a strong oscillator has a large amplitude and a fast amplitude relaxation, and is self-sustained. And the weak oscillator is more at a small amplitude, is closer to the Hopf bifurcation and might have a slower amplitude relaxation rate. So it's a bit vague, but helpful for physiological interpretations. But here's a nice study of Steve Brown, and he has shown that, say, chronotypes are related to the intrinsic period. We measured from skin fibroblasts the intrinsic period of, say, poor people that were extreme chronotypes, and indeed there was a clear correlation between period and chronotype. And there are better data from the Boston teams, but it's also plausible. So if you have a short delay, short period, then you are typically earlier. That's why I don't show too many other data. This is some lizard data. So here you drive lizards with different Zeitgeber strengths, so 0.9 degrees up to 7.2 degrees. And you see the entrainment range of such a weak Zeitgeber is small, plus minus half an hour. The entrainment range of 7 degrees Zeitgeber is larger, plus minus 1 hour or even more. You see also the phase is changing a lot, but the slope is far larger, say, for weak Zeitgebers than for strong Zeitgebers. So you see some features that are features of driven oscillators. And I don't want to repeat now Arnold tongues and saddle node bifurcations and resonance curves in detail, but I can promise that everything is in an Arnold tongue. So here you see an entrainment region of a model oscillator, and you see for strong oscillator like mammals, you have a small entrainment range. For weak oscillator like Neurospora, you have a huge entrainment range. At the same time, you see the phases of entrainment go from minus 6 hours to plus 6 hours. And this is related to the experts to saddle node bifurcations of limit cycles. So it's also well understood. And at the end, we learn that if you are a mammal, then a small change in periods leads to a large change in phases. If you are a Neurospora, then say a change of a period of 1 hour gives you also a phase change of 1 hour. So that's why you learn from simple simulations of the driven Poincare oscillators. You can learn a lot about phases and amplitudes and entrainment and entrainment range. And the summary, again, a vague summary is if you have sloppy oscillators, then you have a wide entrainment range, a resetting phase response curve, not discussed today, and a stable entrainment phase. If you have coupled networks, like in our SCN, then you have a narrow entrainment range, small PRCs, plus minus 2 hours, but a quite flexible entrainment phase. And this, without any theory, has been shown by Aschoff in 1978. So he studied the entrainment range and the phase of entrainment for vertebrates. And you see a very narrow entrainment range, about plus minus 2 hours. These are all the data points. But you see a strong variation of the phase of the chronotype. But in plants and unicellular organisms without too much coupling, we have a wide entrainment range, plus minus 6 hours and more. But this means that the phase changes are minor. That's why a lot of data can be at the end explained with this figure. It's just another experimental evidence that the strength of oscillators matter for entrainment and for the phase as well. And now I can give you the answer to the question. The SCN is a strong oscillator due to coupling, and this implies that the amplitude is large. The entrainment range is small, but the phases can change by many hours. And so everything is consistent with oscillator theory. But I fully appreciate that if you are not a physicist and you never heard about the saddle node bifurcations and Arnold tongues, then it's hard to understand it quantitatively. But still, I might have convinced you that it's not rocket science. So it's really basic oscillator theory that can provide a lot of answers backed up by many data. And this leads to the final summary. So it's helpful to distinguish strong and weak oscillators. And if you have appropriate models, then you can predict many, many properties of driven oscillators. And the theory explained the wide range of chronotypes. And this was known to Rutger Wever, an engineer in 1964. It's not my result, but it was almost forgotten. Since in modern times, all the chronobiologists, they only care about genes and clock genes and knockouts, and they have forgotten the good old theory of amplitudes, phases, synchronization, and entrainment. And that's why it's sometimes helpful for theoreticians to remind the good old days of when organismic rhythms were known and the oscillator theory was advanced. And in mammals, say robust oscillators imply flexible phases or widespread of chronotypes. Okay, now time is over. So there are also medical applications. So we have clocks in every tissue, and you can apply the whole concept to cancer chronotherapy. So if our body is rhythmic, then also the effect of drugs might be different during the day and during the night. And this has been shown by Francis Levy. It's helpful to give drugs at the right time of the day. And together with Samuel Bernard from McGill, originally, we simulated chronotherapy and found out, yes, indeed, the rhythmic treatment is helpful, at least in the computational model. And we could even make predictions. So for fast-growing tumor, it might be good to drive the drugs with 29 hours as a rhythm. Might be better than the typical 24-hour rhythm treatment. But with programmable pumps, it's possible. This is a bit crazy since it's a computational model and related to therapy, but it's perhaps the first step. If you have a reasonable model, then you can at least predict something that is not intuitive. And at the end, the secret is clear. If you drive a rhythmic tumor with 24 hours, then it's risky. So if you have the wrong phase, then you have more, say, harm than gain. But if you drive with a reasonable period that is not close to the resonance, then you might get the better treatment. This is a bit speculative, but at least the resonances are confirmed in mouse models. So if you have the wrong treatment by 4 hours, then you kill many more mouse than if you have the best period. So this is not totally crazy, at least in mouse models. And I summarize with a slide that is showing, yes, everything is rhythmic in our body. And even some features like cell division, heart rate oscillate. And it's also related to medical issues. There's a very clear link to mood disorders. So if you have a poor clock, then the likelihood of sleep disorders and of depressions is much higher. And there is even a modulating issue. If you have an intact clock, then the likelihood of breast cancer is a bit lower. So clock is not an oncogene or tumor suppressor, but it's modulating many diseases. And that's why it's good to know that we have a rhythm in our body just to optimize, say, treatment or even behavior. And my take-home message to all of you is you should be outside at least half an hour per day and to walk a bit and this helps a lot to sleep well. And then you should avoid bright light close to midnight. And then it's another support to sleep well. And you can, even if you are an early bird, then you can change it a bit by late light. Or if you're a night owl, then you have to go out early. Then you can switch your clock a bit, and then you can say you are less tired in the morning if you have an exam. Also. Okay, we are not slaves of our clock. We can influence it, and it's easy to influence it by light. By melatonin, it's an alternative, and so on. So this was the summary, graphical summary, and thanks for your attention, even a few more minutes. And I'm happy to go on with questions if you have some.
Thank you so much. Are there other questions for Dr. Herzel. So there's one in the chat.
Hans Otto, it's Mike Mackey here. Can you hear me?
Yes, I can hear you.
Hans Otto, I'm sorry, not Hans Otto, Hans Peter. I thought the issue about chronotherapy, I thought Francis Levy had tried a number of things and had had great difficulty treating patients. And I think at one point he discovered that with colorectal cancers, either men or women did not do well with the chronotherapy, but the other sex was slightly better. I don't remember the details. Have things progressed from that point? This is a number of years ago that I remember talking to him.
So the story is interesting. So indeed, Francis Levy was performing a clinical study, a big clinical study with colorectal cancer. Unfortunately, he was allowed only to treat stage 3 colorectal cancer. And then the median survival time is something like 15 months. No? And you are right, you remember correctly, say men survived a few months longer, no? And women died a few months earlier. That's why, from a pure survival point of view, it was disappointing. And from a scientific point of view, it's interesting, since also these women were old, so beyond the menopause. That's why hormones are not the main driver. Why women react differently? This is an interesting scientific question. We have no answer yet. No? In the meantime, say Francis Levy is now retired, so he's not making other clinical studies, no? But he confirmed that the side effects were reduced a lot. So even if people died also after 1 or 2 years, the reduction side effects were immense. So the level of drugs to reduce the tumor were much less harmful. So that's why there was, even in this study, a gain, but not in lifetime, but in side effects. In the meantime, there were a couple of studies, not by Francis Levy, but independently. For example, in glioblastoma, some people have shown that if you knock down BML1, then stem cells cannot grow anymore well, and that's why the relapse is less likely in glioblastoma, a terrible brain tumor. And there's another study of melanoma, and the current therapy is immune therapy. And some people found out that if you give immune therapy in the morning, this is much better than giving it in the evening. So there are even clinical studies from non-say chronobiologists that are convincing. These are snapshots, but it's plausible, and there are at least some snapshots where some advantage of chronotherapy has been proven. So it's not hopeless, but still a long way to go.
Okay. And thank you very much. Excellent talk.
Thanks.
There's a question in the chat. Do you have any insights on the coupling strength and feedback delay distributions in the oscillator networks?
So I try to read also the chat. Let's start with coupling strengths. So we are aware that SCN neurons are coupled seriously. So I might ask the theoreticians, how do you define weak, medium, and strong coupling? So Michael or Andre, do you have an idea how to distinguish weak coupling from medium coupling from strong coupling?
I'll leave that one to Andre.
So my answer was published by Art Winfrey already. So he's saying if the effect on the oscillator is, say, below 5% or 10% on amplitude and period, then I term it weak. And then you might go to phase oscillators. Ne? If the effect is, say, 50%, ne, then it's medium. Then you might go even to the singularity of art infinity. Ne? If you dictate the period and amplitude by the external driver, ne, then it's strong. So that's why the amount of effect, if you turn off or on the coupling, and then you can monitor the effects. Ne? And if the effects are a few percent, then it's weak. Ne? If they are more than 50%, then it's strong. So this is, say, our preliminary definition of weak and strong coupling. And this is related to phase response curves, since it's also related to phase resetting and so on. Now to the real question. So I'm convinced that the coupling in the SCN is medium. So it's strong enough to synchronize safely, but it's not strong enough to dictate the period. Since the period distribution is centered around 24 hours, 20 plus minus, or 24 plus minus 2 hours, but the average period of a synchronized slice is also around 24 hours. This is one argument that the coupling is not dictating the period and the amplitude. Also, the amplitudes are not changed too much. No? No? Now, the question is, are other cells in our body coupled with each other? And the answer for fibroblasts is perhaps a little bit. So there are some indications by recent papers of Anna Finger and Achim Kramer that fibroblasts in high densities are coupled a bit. And the coupling agent is even found. It was something related with TGF-beta, if I remember correctly. Uli Schiebler from Switzerland, he found that liver cells are also coupled seriously. So if you kill the SCN, if you have dark situations, then still the liver ticks with its own period over many weeks. So that's why there's, say, reasonable evidence that also liver cells, hepatocytes, are coupled to each other. And that's the current stage. So this is the intra-organ coupling. So it's weak to medium. There is, of course, also coupling between organs, and this is mainly by glucocorticoids, by body temperature, by vasopressin, and this is diverse. So you have a strong coupling of pancreatic islets to the liver, or of adrenal gland to the liver due to glucocorticoids and so on. But this is too complicated for a modeler. So there you need really strong physiology and biochemistry to quantify this coupling. So there was another part of the question regarding distributions of delays. So I know the distribution of periods, as I mentioned, so it's plus minus 4 hours in single cells. And this might indicate the distribution of delays. But since single cells are so noisy, I can't quantify delays in single cells. I can only guess from the action of kinases or of, say, nuclear import that there is a distribution. The J. Kim from Korea, he has some data about the delay of nuclear import of period 2. And his claim is that local switches are helpful to reduce the noise of the noise. So there's a recent paper in iScience of J. Kim. This is a bit related to the distribution of delays and to the role of switches to get still good synchrony. So J. Kim, iScience, I guess last year.
Question from Katie, perhaps?
Yes.
Yes, hi again. Thanks for the talk. It was very interesting. I just have a quick, conceptual question. More like, what could you say of the circadian rhythm in blind people? Like, can the skin or other organs detect the sunlight, and how good it would be, and would the SCN still be considered a strong oscillator in these people for the other cells? Yeah, that's it.
So, I guess the SCN is still strong, but the Zeitgeber is very, very weak.
Okay.
And that's why half of the blind people are typically free running. So sometimes they sleep at night, but say 2 weeks later they sleep during the day, since the intrinsic period is not the same as the extrinsic period. Of course, they can be synchronized by meals. This is a second strong zeitgeber. They can be synchronized by drugs like melatonin or glucocorticoids, or by social life, let's say by alarm clocks. And so it's not hopeless, but they need some external cues to be synchronized. So they are probably strong oscillators, but the zeitgeber is naturally very weak. In
Okay.
birds, say there is direct light input to the pineal gland. In mammals, there's nothing known about other strong inputs. There are some debates that the retina is driven by light, but but the light driven knee oscillations was a fake. It was an artifact. So if you find a high ranked paper that you can synchronize clock in the knee by light, but this was not confirmed. Yeah, so we need, say, light input to the SCN to drive our rhythms normally. But meals can replace it, or social cues and or drugs.
Okay. Thank you.
Last chance for a question, if there's any others.
Is there something in the chat left?
Nothing in the chat.
I don't think so. Yeah.
Okay.
Listen, thank you so much, and that was a really stimulating talk. Lots of questions, and a real pleasure to hear your research. Thank you.
And for me, it was a pleasure to see some of you again and say bye bye and all the best.